Steve's lab. And also, many of you might think I'm just here for the practical jokes and the bad jokes, actually. So I'm going to show you a little bit of what we've been doing here at the college. We can be found in a number of labs. We have a toxicology lab upstairs on the second floor, 227E, where we do most of our extractions. The new environmental chemistry lab, which most of you might have had a chance to go in and see. And then we also have the population dynamics lab down in 149. Steve is also the PI of C-Image, which is a very large consortium of about 17 institutions to help answer the one question about the Deepwater Horizon, which is what I'm going to present to you today. Maybe. So first, I'd like to introduce you to his lab. He has a number of students working on a wide range of projects that range from population dynamics, growth, if I could learn, I can't figure out how to work this. Population demographics and golden tilefish. We have a number of students in this room that have been working on the toxicology portion of Deepwater Horizon. We have students, Alex here, that's been working on sea mapping, and some other students working on environmental indices from the Deepwater Horizon. But this is to show you the range of projects that he has. But you might notice the one common denominator. This is Steve, by the way, for any of you that don't know him. But basically, all the research is just about fishing. So you must like fishing. So again, we're going to talk about the Deepwater Horizon. By the way, this is from Steve, Deepwater Horizon. And I don't know if he's a Marky Mark and the Funky Bunch fan or the Mark Wahlberg fan. Maybe some of you are too young to know who Marky Mark is. Underwear mom. Anyway, so the Deepwater Horizon, as many of you may know, was a very large, one of the largest spills in history that hit the Gulf of Mexico, spilling about 4.9 billion gallons and covering thousands of miles of shoreline, impacted surface waters down to the benthos. So our job has been tasked with how do we measure impacts and recovery in the Gulf of Mexico, which is a semi-enclosed basin, highly productive, highly diverse, and highly dynamic. So how do you measure that when there's very limited baseline data, which is very surprising considering the oil and gas activities in the Gulf of Mexico have been here for over 7 decades. In addition, there have been over 343 oil spills off the outer continental shelf of the Gulf of Mexico. Since 2010, when Deepwater Horizon occurred, there's been about an additional 9 oil spills. We have the Taylor Energy well that's been leaking since 2004. In addition, we have riverine input, atmospheric deposition, vessel traffic. So there's a number of inputs, not just from this major oil spill. And here are some interesting statistics that I thought were quite staggering. There's over 7,000 platforms in the Gulf. There's over 27,000 abandoned wells, and there's over 25,000 miles of pipelines. This here is a graph of the black dots are all the oil wells in the Gulf of Mexico, and the red dots are the natural oil seeps. We have Deepwater Horizon here, and then we have the Ixtoc oil spill that occurred back in 1979, 1980, which was also quite large. The other problem we have is traditionally or historically, everybody didn't really pay attention to PAHs, which is the very toxic component in oil, but they kind of disregarded, which I think is part of the reason why we have no baseline data in fish, is because it's always been assumed that fish can metabolize them and they don't accumulate them, which is true. Their metabolism can metabolize them and excrete them. They use the same CP54 system that we do, so they're very efficient at metabolizing them. So nobody's ever really kept monitoring the levels in these fish, even though we have chronic impacts from the oil and gas activity in the Gulf of Mexico. So I'm going to present some of the data from our students, the students in Steve's lab that have been studying this for the past few years. This is a map of all of our longline surveys that we have done in the Gulf of Mexico since 2011. We've covered over 359 longline stations, set about 150,000 hooks, sampled over 15,000 fish, number of over 200 unique species. A number of these sites since 2011 were repeat stations in the north central region of the Gulf to try to monitor spatial and time trends. We added this component to compare the Ixtoc oil spill to the Deepwater Horizon oil spill. So this is Susan Snyder's research here, who's sitting in the back. She actually is finally defending. Just kidding, Susan. Please come see her defense. Or you could just look at this. No, just kidding. This is just one chapter. So Susan Snyder has been studying golden tilefish in the Gulf of Mexico for the past number of years. The reason she chose those are demersal species that are benthic. They're known to burrow. So the assumption was that they were going to have high levels because PAHs are hydrophobic. They tend to bind to particles and sink and fall out of the water. They're also lipophilic, so they are going to bind to lipids in any fatty tissues, and that's where we're going to find them accumulating. So Susan looked at these fish since 2012 in the North Central. This is her Time Trend series paper that's actually in press and should be out probably next week or so, a week or so. Sorry, this is hard to see. Here, she's looking at condition factors over time. Here we have all the data clumped together, and then she separated them out by sex. But overall, what we're seeing is a decline in condition factor, and this is an indice of health. So over time, this tile fish from 2012 and 2017 have been declining. She's also found some increasing, 178% increase in PAHs over time. Initially, the first few years after the oil spill, there was a decline. So this brings up the question, when you're measuring recovery, how long do you measure that for? 1 year, 2 years, 3 years? But I think this data argues the point that we need to continue monitoring, especially with the continued oil and gas impacts in the Gulf. Additionally, she looked at liver PAHs over time. However, these tend to maintain, they were stable over time, which is interesting. With the increased exposure, we might have expected increased accumulation in the liver. Biliary PAHs are really indicative of short-term exposure, whereas the liver PAHs are long-term chronic exposure. So if you look at tilefish in the Northern Gulf, they are still efficiently metabolizing and excreting them out of the system and not accumulating them in their fatty tissues, such as the liver. I think she's looked at muscle, but we don't expect high levels in the muscle because it's pretty lean muscle or tissues comparative to the liver. So if we move on to Christina Deeks' work, Christina Deeks is a PhD student here. You might see her, she's often covered in blood and carrying a meat cleaver. But don't be afraid, it's just after the fish. But she's working right now over in the health department at FWRI. And her main research has been novel indicators of oil exposure and health effects, looking at the chronic sublethal impacts of oil. As Alex pointed out earlier, there's very visual impacts of oil spills like mortality, but what about the sublethal impacts that you don't see? And Christina Deeks has been working on that as well, looking at golden tilefish in the yellow, red snapper in the red, and she's collected samples from around the Gulf. She basically looks at the blood and some cellular mechanisms. Here's just a couple of the things that she's looking at, erythrocytes and DUD, or SUD, I'm sorry, to look at the immune function in fish. And so what she's finding, and this is Here is a regional comparison between Campeche Bay, which is down here in Mexico, the north central region, and the northwest. And what she's finding is that the golden tilefish, compared to the red snapper, has much higher genotoxic exposure in those fish, which can induce some abnormalities later on down the road. And I'm probably completely butchering her PhD, but she's looking at the immune function of the fish and what kind of impacts it has on those fish. And the north central region, which has been impacted for quite a long time, is showing some induced changes. Over here is a measure of the antioxidant, and again, also I should point out that the red snapper is much higher than much lower than the golden tilefish. Here is another measure of antioxidant that can be increased due to exposure to PAHs in fish. And again, the north central region for golden tilefish and for that matter red snapper are all higher than any other region. If we look at hakes, this is another paper that will be coming out here soon from a master's student, Rachel, who was looking at hakes, another demersal fish in the Gulf of Mexico. And she also noticed in this species, however, she shows a decline in biliary exposures, an 18% decline. However, liver levels are increasing 119%. In this particular species. And again, her data set stops at 2015. Here's some of the work that I've been working on, and we looked at 9 species of grouper from around the Gulf. This is probably about 500 fish or so. And what's interesting here, we were able to separate out each species by the way they accumulate these PAHs in their liver. We have some of the deep water Warsaw, yellow edge, and some of the shallow water species over here. Here I'm also seeing a decline. This is condition factor on the bottom by year, percent lipids up here. And what we see is a decline the first few years after DWH in both lipid and condition factor, and then we start seeing increases again. So is this where they're starting to recover from the initial impacts of Deepwater Horizon, or is there some other mechanism that we don't know about? Again, over here we have biliary PAHs on the top, liver PAHs on the bottom. Over time, this is just from the north central region, I should point out, which is these fish up here. But we also see the same trends that we do with increasing liver concentrations, initial decline in exposure, and then we start seeing an increase again. So the question is, where's these increases coming from? And they all are occurring. We see a blip here, and then in 2017 we jump up quite high. So is that from resuspension events that we're seeing, or is that from continued leaking and continued spills? Let's see. And Maddy Schwab is a master's student. Also, I don't see her here, but she's now looking at the pelagic realm. So in 2018, we did another cruise, and we just did some pelagic longlining right around the, very close to the Deepwater Horizon spill, and she compared those to some other pelagics that we collected Gulf-wide. And what she found was yellowfin tuna were had some exceptionally high levels of biliary naphthalene, higher than golden tilefish, which before now had had the highest levels that we had found, which tended to make sense considered they were demersal. So this here kind of illustrates that it may not be lifestyle and life history styles that are influencing these levels. It's exposure, swim speed, etc. That's right. And this is going to be another offset of her research. I don't know if she knows it. If she's not here, that's good. So what's interesting is, again, we're looking at the plume came down here, but she's looking at these pelagic species up here, which feed off of some of these mesopelagic fish. So what we want to do is she's been working with extracting the tissues in the pelagic species and the stomach contents to be able to look at the contribution of you know, the mesopelagic influence and how the trophic transfer up the food chain, the PAHs are going. We've also been working with Maya's lab as well to identify some of these partially eaten stomach contents. So. And then on to Bridget, who is here. So Bridget is a master's student also, and she's looking at some additional anthropogenic contaminants, some legacy compounds, PCBs and pesticides, as well as PAHs and snapper off the coast of Cuba. And she's found some interesting results. She's finding some basically Havanas here, and then we move west. And so what she's finding is Havana is 44. We're finding some declines in PCBs with distance from industrialized areas. Pesticides and PAHs don't really show the same spatial gradient, but she's not completely done with her data set, and she's also going to now compare, because why not throw the whole kitchen sink at them, compare that to snappers from around the Gulf. So putting it all together, this is some work that I've been doing, is trying to compile all the biliary data that we've collected over the Gulf over since 2011. And so what we're finding is that on the top graph plot here, this is biliary naphthalene throughout the Gulf. And hot spots are obviously the Deepwater horizon, but we also have them by major ports and harbors and tributaries. Very low around Ixtoc, which was relatively surprising since there was a very large oil spill. But again, this might be where should we use this as a proxy for recovery? When this area begins to decline in levels, will we be at full recovery? The bottom plot is biliary benzoate pyrene, shows a little different profiles, but we do still see some hot spots around the Mississippi River Delta and major river inputs. BAP or benzoate pyrene is more of an anthropogenic high molecular weight compound that we associate with more petrogenic, pyrogenic type sources. This plot over here is just showing you a few of the species over time and how, so it's the earliest year, 2012, going up 2017 for each of these species. Most of them are increasing. We do see some dips, but most of them are increasing exposure over time. So why does this all matter? Well, with the oil industry and gas activity continuing to expand, they're moving further and further east towards the moratorium area, and when that is lifted in 2022, what types of things are we going to see? With the fish, with the chronic levels that we're seeing as they're increasing over time, when is that point that we're going to start tipping the... This is just a graph showing how we're going deeper and deeper waters for drilling over time. So what are the impacts going to be at those deeper and deeper depths? So as a result of all the research since 2011, we do have a couple books that just came out. So if you're interested in more information on that. And any questions?